Internal structure of dicot root. This topic explains the internal structure of a dicot root. Let us observe a sunflower plant, which is a dicot plant. It is a herbaceous plant with a green, slender stem. The leaves show a reticulate venation. If we look at its root, it appears branched. On taking the transverse section of the root, several tissues can be seen. Let us study each tissue one by one. The outermost layer is the epiblema, which consists of thin-walled, compactly arranged cells. There is no cuticle on the epiblema. A few cells of this layer protrude as the outgrowth called the root hair. Epiblema is thus absorptive and protective in function. The next layer is the cortex. The cortex is composed of large, thin-walled, spherical or oval parenchyma cells. Large intercellular spaces are present, which help in the diffusion of water in the cortical cell. The endodermis consists of a single layer of oval cells that have thick deposition of suberin over them, which is characteristic of endodermal cells and is known as casparian strip. Next is the single layer of the pericycle, which is made up of the thin-walled parenchyma cells. Embedded in the parenchyma cells of the ground tissue are the vascular bundles consisting of the xylem and the phloem. The xylem and the phloem lie at different radii. The xylem has two sections. The protoxylem is towards the periphery and metaxylem occupies the inner position. This arrangement is known as X-arch. The xylem and the phloem are separated from each other by a few parenchyma cells called conjunctive tissue, which is a part of the ground tissue. A quick recap shows that a section of a sunflower root shows the outermost layer called the epiblema, and then lies the cortex. The next layer is the endodermis and pericycle. Xylem and phloem lie inside. They are surrounded by the ground tissue. Ground tissue between the xylem and phloem is the conjunctive tissue.